that's water splitting. Any questions about that? Okay. Electrons move from PS2 to the cytochrome B6F complex. And the transporter here is quinone. All right. And remember, quinone picked up 2 protons from the stroma, bound them, and then it's going to transfer 2 electrons simultaneously. Okay. And going to donate 2 protons into the thylakoid lumen. All right. And the way the 2 electron transfer works is that one goes to cytochrome B, the other goes to a risky iron cofactor that transfers it to cytochrome F. Okay.
Why'd you call it risky?
I think it's named after the guy that discovered it.
Oh.
So it's actually called risky.
Yeah.
Sorry.
So it's spelled like this.
That was one of the better questions of the semester so far.
And I'm not 100% sure if it's really named after a guy, but that doesn't, I mean, it's not another word that makes any biological sense.
Okay.
But this is what they're called, and so it's an iron molecule surrounded by 4 sulfur atoms. And what it means, what this means is that the iron can very easily go back and forth between +2 and +3 oxidation states. And then cytochrome F will donate one of these electrons to plastocyanin, which is a 1 electron carrier, which makes the rest of electron transport nice and simple. What's the problem?
No.
What have I left out?
What happens to the one that was the cytochrome B?
Exactly. What happens to the cytochrome B electron? So what's going to happen is that this is going to donate an electron to quinone, making a semi-quinone. Okay. What do you think? What do you think happens?
Positively charged.
All right. The quinone molecule, how many electrons does quinone accept?
1.
2.
2. All right, because we've got quinone, semiquinone, and then PQH2, plastic quinone. All right. So what's going to happen is that, if we've got a picture. The semiquinone will stay here until it receives a second electron, and then it departs. What do you suppose it does when it departs? Guess what? Picks up 2 more protons. We've got QH2, and this can funnel into the cycle again. So essentially what's happening is that quinone donates 2 electrons. Only one of those 2 electrons moves through in a linear pathway. The other electron generates semiquinone and then plastiquinone, picking up protons, and it feeds back in. So it's not an efficient system by any stretch of the imagination, but it's one that works.
Okay, so PQH2 can only carry 2 electrons, right?
So quinone, just Q, can pick up, let's make sure we're Q star minus.
Because it said, because it has like, it already has 1 electron there, then at the very top, when from cytochrome B, it goes into semiquinone. Does it lose that electron before it picks up those 2 H plus protons?
So what happens, here, let's go back 1 slide. All right. So in order for quinone to go to plastic quinone, it must pick up 2 electrons. So it picks up 1 electron, going from quinone to semiquinone.
Right.
That's the same as Q.
So that's picking up 1 electron. And then the semiquinone here picks up a 2nd electron, in which case it's going to pick up 2 protons from the stroma. We've regenerated QH2, and this is going to funnel back in at this site of cytochrome B6F.
Oh, yeah, earlier, oh, so when the QH2 goes in, 1 electron goes one way and the other goes the other way, is it ever 2 go one way?
No. In order for it to happen, one gets put into the iron risky sulfur protein, which goes to cytochrome F. The other one goes to cytochrome B. And you never get 2 funneled simultaneously the same way. Genza.
You mean the Q produced by the QH2 will transfer to B6F?
So this plastiquinone here, all right, is identical structurally to this one, and it's just going to get shuttled back in.
Can I go to the?
Sure.
I thought you should say this Q will transfer to here, so the E will get back here, so it's a circle. This what you mean?
So
This Q will get here, then get back.
So the thing to do is to not think about this specific one going to here. Think about having a pool of quinone circulating all throughout this membrane, okay? And so when this plastiquinone donates both electrons here, generating quinone, all right, this quinone can accept electrons either from PS2, so it can go back and accept more from here, or it can go up to cytochrome B and receive electrons from cytochrome B.
Then
But in order
come back to-
for it to regenerate QH2 from Q, it must accept 2 electrons.
Yeah, but here only produce 1.
Yes, so it's going to accept them 1 at a time. So you have to have 2 events, and that's why I had 2 slides, right? This one here, we get quinone reduced to semiquinone, and the semiquinone, when it receives a 2nd electron, it generates plastiquinone.
And it's moving the hydrogens just from the thylakoid and blue lumen.
Yes.
Essentially.
Yes. So we're concentrating protons in the thylakoid loop. Thank you.
The Q and the semiquinone with the dot, that's just the same as QA and QB in the previous slide, right?
Yes.
Okay.
But it's differentiated because it's quinone, semiquinone, and plastiquinone that's not associated with the reaction center at that moment.
Okay.
So that's the difference essentially.
Okay. Thank you.
Now, the beautiful thing with this is that I fried your brains, but everything else for the rest of the day is easy. Okay?
Is quinone going to pile up in this structure? Because for one quinone to go back as plastoquinone, two has to come in.
Say that again?
Because only half the electron goes back to the quinone. The quinone coming in should be twice as the quinone coming back out. So will quinone like pile up in this structure?
So the answer to that question is no. And the reason is that over the distances that we're talking about, quinone can move into and out of cytochrome B6F complex and throughout the thylakoid membrane in microseconds. The distances are very, very small, and the rate of the quinone, again, is a very small molecule. So it diffuses, in the scale of a thylakoid, it diffuses extremely fast. So it's never going to accumulate anywhere. And it appears that the pool of quinone is never ever limiting for photosynthesis. There are other things that are limiting for photosynthesis. Typically it's phosphate for ATP synthesis. So that's almost always where the blockage is. And when we're done with photosynthesis and done with carbon fixation, we're going to talk about a bunch of ecological considerations in the way plants adapt to high light conditions, which is actually fairly easy for them to do. But what's rather extraordinary is how they adapt to sunflex. And I'm sure you guys have noticed that there'll be spots on the ground that receive full sunlight, and then seconds later, because the leaves have rustled, that spot will be shaded again. And the amazing thing about plants is that they're able to receive 2 or 3 seconds of full sunlight. It doesn't fry the chloroplasts, but then they go back into a light environment that has 1,000-fold less light. So typically full sunlight, we talk about 2,000 microeinsteins, and you know how bright full sunlight is. Christine, do you have any trouble seeing in the woods in the shade?
Sometimes.
Sometimes?
Yeah. Then your eyes get adjusted to it, though.
Yeah, usually your eyes will adjust pretty fast, right?
Yeah.
So,
I mean, if you walk right in from the bright sun to a shaded forest, then you
and
can't.
your
Right.
pupils will change their dilation and open up so that they allow more light in. Typically, when you shine a light in your eyes, your pupils go to little tiny pinpoints. So the amazing thing is that that's typically at 10 microeinsteins. 200-fold difference. And our eyes are perfectly comfortable adapting to that. And the amazing thing is that plants adapt equally well. So we'll talk about sunflex and why they're interesting and important. Okay. Electrons either directly shuttle into cytochrome F, or they do this little tango dance and shuttle into cytochrome F. But then the electron acceptor from cytochrome F is plasticine. And this is a transporter that moves along the periphery of the membrane and will donate electrons to PS1. All right, and so we've got plasticinin donating an electron to a reaction center, And then there are a couple intermediate states that exist, including another iron risky sulfur protein, 2 of them actually. And eventually the electron is donated to ferredoxin. Okay. And ferredoxin, so the only point I haven't mentioned is that plasticine will typically dock and interact with PS1, and it won't donate the electron until an electron has been ejected from our P700 reaction center, and this is P700 plus. Then the electron gets donated. So essentially P700 needs to lose an electron through excitation before the electron from plasticine moves in to replace it. So this plasticine can remain docked with PS1 for milliseconds in a light environment and hours in darkness. It just depends. Chenza.
So in this process, it will cause a photon,
Excuse me?
the light, the photon.
It requires a
Process
photon.
Oh, yeah. The removal of the electron from P700 requires a photon of light, correct, absolutely. So that electron must be excited by a photon before it can be, before it's ejected from the reaction center.
It will cause one photon.
One photon, just one photon here, one photon for PS2. Thank you.
This might be a silly question, but does light from like a fluorescent light bulb matter versus like sunlight? If they just are producing the same wavelength?
So in terms of the chloroplast, no, it doesn't really matter. Fluorescent as someone that, for our grower, it's important because plants are adapted to a full natural spectrum of visible light. However, almost all grow lights have deficiencies in terms of the natural spectrum. Jacob, do you want to hazard a guess at how much a light bulb costs that mimics the natural spectrum of light?
I know
They have those?
light systems can go up to like 10 grand easily.
So the fluorescent light bulbs that are in this room, each bulb costs 20 cents. And the reason I know this is that those are the fluorescent bulbs I typically use to grow my plants.
Really?
So a full spectrum fluorescent light that has an interesting combination of heavy metals instead of just neon inside the tube, $50. And people buy them because plants have weird phenotypes when you grow them under almost exclusively blue light.
Why do you think that is?
Why do they grow differently?
Yeah. Well, like the phenotype you said.
We'll spend a week discussing your question.
Okay.
Can you wait a
Thanks,
month?
Dalton.
Yeah, I can wait.
Okay, because it's not really a simple answer. There's a lot going on there. But for plant growth and morphology, the spectrum is very important. For photosynthesis, all that matters is that we have a photon of light that can be absorbed by the antenna complex. And the most important thing is the intensity of that photon. So the answer to your question is, yeah, it matters a great deal for growth and morphology.
But
For
not
photosynthesis,